Welcome, everyone. We are genuinely thrilled to have you sitting right here at the table with us today. Over our last few deep dives, we've been meticulously mapping out the raw, intricate machinery of human reproduction.
The microscopic structures, the cellular divisions.
Right, exactly. The hormonal cascades, the sheer staggering biological engineering of how life basically goes about creating more life. But today, we're stepping back from that microscopic view.
We are pulling the camera way out.
Way out. We are looking at the macro picture. We are asking the fundamental question, what happens when that perfectly tuned biological machinery interacts with human society, with our complex emotions, and with the raw, undeniable mathematics of population growth?
And that is exactly what we're going to explore today.
Yes, as we dive into reproductive health, which is Chapter 4 of your biology curriculum. And listen, we aren't just here to recite facts at you. We are going to map this out to help you truly understand the high-yield concepts.
The biological mechanisms driving them.
And those hidden, tricky traps that examiners just absolutely love to use to test your true comprehension.
What's fascinating here is that when we talk about reproduction in the wild, the broader natural world, it is a completely unchecked biological drive.
It's ruthless.
It really is. It is a fundamental, almost brutal imperative for any species to survive, to multiply, and to pass on genetic material, often regardless of the immediate environment. But human society operates on entirely different principles.
Because we have limits.
Exactly. We exist within the strict bounds of finite resources: food, space, healthcare, physical infrastructure. So this topic of reproductive health isn't just a localized chapter about anatomy or pathology.
It's the ultimate bridge between biology and sociology.
Precisely. It exists to solve a massive existential problem. How do we effectively manage our incredibly powerful biological imperatives while simultaneously maintaining a balanced, healthy, and thriving society?
It's about the survival and the quality of life of our entire species, really.
It is. And that is the analytical mindset we need to equip you with today.
That is exactly the framework we need. It makes everything we are about to study feel intensely urgent and relevant. Our mission for you in this deep dive is to extract the absolute essence of this topic. You are our third partner in this dynamic study session today.
They're building this together.
Together, we are going to build a comprehensive mental map, highlight the specific high-yield concepts that consistently appear in your Neet exams, and unpack the actual science behind why these systems work the way they do. So let's get right into the blueprint.
They'll do it.
Okay, let's unpack this starting with the very definition of the term itself. Because if you read the World Health Organization's definition, it fundamentally reorients how you think about medicine.
It's not just the absence of disease.
Right. The WHO states that reproductive health is a complete mental map built on 4 distinct, unshakable pillars: physical, emotional, behavioral, and social well-being.
And let's build a prior knowledge bridge right here for you. Think back to Chapter 3, where we painstakingly detail the anatomical structures.
The internal architecture of the testes.
The histology of the ovaries, the ciliated epithelium of the fallopian tubes, the secretory functions of the accessory glands. You could theoretically have a society where every single individual has physically and functionally flawless organs.
Anatomically textbook perfect.
Exactly perfect. However, if you do not have normal, healthy emotional and behavioral interactions among those individuals in all sex-related aspects, that society is simply not reproductively healthy.
The WHO definition is profound because it forces clinicians and students alike to look beyond the microscope.
It asserts that the psychological state, the community dynamics, and the behavioral patterns are just as vital to the concept of reproduction as the gametes themselves.
It is a much more holistic, integrated way to view human physiology. And what is particularly interesting to me, looking at the historical context provided in the source material, is how early on this macro-level understanding was officially recognized, particularly in India.
A very proud moment in global public health history.
India was actually amongst the very first countries in the entire world to initiate national action plans for this. They launched their family planning programs all the way back in 1951.
1951.
Just a few years post-independence. For a newly formed government to recognize that reproductive health was a critical foundational social goal alongside agriculture and industry, it's quite remarkable.
It really is.
Today, you will see all of these initiatives operating under a broader, much more comprehensive umbrella known as the RCH programs.
Reproductive and child health care. That 1951 milestone is a highly testable fact. India recognized the impending mathematical reality of population dynamics incredibly early. And this brings us to a fascinating, almost cruel paradox of modern medicine.
The unintended consequences.
If we connect this to the bigger picture, the very medical successes we achieved in the 20th century actually created a looming sociological crisis. Think about what was happening. We had better general awareness.
The massive increase in medically assisted deliveries.
Institutional deliveries, yes, and vastly improved postnatal care through antibiotics and better hygiene. All of these incredible scientific advancements led to a rapid, steep, and sustained decline in the maternal mortality rate, or MMR.
And the infant mortality rate, the IMR. We were doing exactly what medicine is designed to do. We were saving lives.
But the mathematical irony is staggering. Because fewer mothers were dying in childbirth, because fewer infants were succumbing to early infections, and because general health care improved across the board.
Millions more people were surviving long enough to reach reproducible age, which leads directly to a massive, unprecedented explosion in the population. I was looking at the demographic math behind this, and the standard analogy perfectly illustrates why this shift was so aggressive.
The bathtub analogy.
Yes. The demographic shift is best understood if you think of it like a massive bathtub with the tap running wide open and the drain unplugged.
The water pouring in from the tap represents the birth rate.
And the water pouring out through the drain represents the death rate. For the vast majority of human history, both that tap and that drain were wide open. High birth rates were grimly balanced by high death rates due to endemic disease, poor maternal care, famine.
A complete lack of medical infrastructure.
Right, so the water level, the total population stayed relatively stable. But with the rapid medical advancements we just outlined, humanity essentially plugged the drain. The MMR and IMR dropped drastically.
However, the biological drive and cultural norm of having many children was still running at full blast.
The inevitable result is an explosive accumulation of water, an explosive impact on the population. By the time the 2011 census report was published, the population growth rate in India was hovering around 2%.
To put that in perspective, that means 20 new individuals were being added per 1,000 people every single year.
Now, a 2% growth rate might not sound like a terrifying number when you hear it completely out of context. 2% sounds like a modest return on a savings account.
But when you apply that 2% compounding growth to a base population of over 1 billion people, the absolute numbers become staggering.
If you apply the biological rule of 70, where you divide 70 by the growth rate to find the doubling time, a 2% growth rate mathematically dictates that the entire population would double in a shockingly short amount of time.
Just about 35 to 36 years.
If your population doubles in 3.5 decades, it leads to an absolute, unavoidable scarcity of the most basic fundamental requirements for human survival. We are talking about severe deficits in food security, a lack of adequate shelter, and insufficient clothing and infrastructure.
The government wasn't just planning for the future. They were forced to take serious, aggressive measures to check this growth rate immediately.
Because absolutely no amount of economic or agricultural progress can outpace a population that is mathematically doubling every 35 years.
Which is exactly why understanding the mechanics of population dynamics is a core pillar of this chapter. But before we transition into the specific strategies society uses to control that tap, we need to address a very specific, highly testable medical procedure.
Oh, this is a big one.
It perfectly illustrates the tension between the physical and social pillars we discussed earlier. This is a major area for exam questions, and examiners love to test your nuanced understanding of it. Let's break down amniocentesis.
Ah, yes, the amniocentesis trap. This is a classic area where students overthink or misread the prompt. Let's look at the actual mechanism first. Functionally, amniocentesis is an incredible diagnostic procedure.
Under ultrasound guidance.
Right. A medical professional inserts a needle through the maternal abdomen to extract a small sample of the amniotic fluid. That's the protective liquid environment that surrounds the developing fetus in the uterine cavity.
Now, this fluid isn't just water.
No, it contains suspended fetal cells that have sloughed off the skin and respiratory tract, along with various dissolved biochemical substances. By extracting this fluid, cytogeneticists can spin down those fetal cells, culture them, and analyze their chromosomes.
A process known as karyotyping.
Exactly. This allows doctors to test for severe genetic disorders. We are talking about identifying chromosomal aneuploidies, like Down syndrome, or genetic mutations leading to hemophilia and sickle cell anemia, ultimately helping to assess the survivability and health of the fetus.
Purely as a piece of medical technology, It is a brilliant diagnostic window into the womb.
It is a phenomenal diagnostic tool, but this raises an important question.
And here is what you must remember for your NEET exams. The examiners will very frequently present you with assertion reasoning questions regarding the statutory ban on amniocentesis. You have to read the exact wording of these questions incredibly carefully.
Because there is a strict statutory ban on this procedure in India.
But the trap lies in identifying exactly why it is banned. The government did not ban amniocentesis because testing for severe genetic disorders is inherently bad or unethical. It is banned strictly and explicitly to prevent its misuse for sex determination.
Exactly. This is where the sociological reality crashes hard into the biological technology. In a society where there has historically been a strong, deeply unfortunate social and cultural preference for male children.
The ability to discover the sex of the fetus early in the pregnancy was being weaponized.
It was leading directly to an increasing and horrific menace of female feticides. The very medical tool designed to ensure the health of a child was being used to commit a massive social evil based on gender bias.
So if you are staring down a multiple choice question asking for the primary reason behind the statutory ban on amniocentesis, the key phrase you must lock onto is to legally check the increasing menace of female feticides.
If an option suggests the ban is to prevent genetic testing or to protect the mother from the physical risks of the needle, it is a distractor. Do not let the examiner trick you.
Perfectly stated. That is a vital, non-negotiable distinction to carry with you into the exam hall. It perfectly encapsulates why reproductive health cannot be studied in a biological vacuum.
Now that we have thoroughly established the scope of the macro problem, the explosive population growth fueled by decreased mortality, and the complex social dynamics surrounding reproduction, we need to rigorously examine the solutions.
If the overarching, hardwired biological drive of a species is to constantly reproduce, how does humanity successfully and safely hit the pause button?
This brings us to the absolute core of population stabilization contraceptive strategies. Okay, let's unpack this massive section. This is where we dive into the art and science of pausing nature.
To set the historical and cultural stage, the government recognized early on that relying solely on medical devices wouldn't work.
They needed a fundamental cultural shift to motivate smaller families. You see this reflected in the widespread media campaigns. The posters and television spots showing a happy, prosperous couple with 2 children, pushing the famous slogan, "Hum do, hamar do,"
which translates to, we too are too.
And in urban centers, many young, working couples have voluntarily gone even further, adopting a one-child norm. Alongside this cultural push, we have statutory laws that play a biological role. The marriageable age is legally set at 18 for females and 21 for males.
By delaying marriage, you are effectively shortening the total fertile window during which a couple is likely to reproduce,
which has a tangible impact on birth rates. But beyond slogans and legal ages, we have the actual physiological, biological hacks that humans use to intentionally prevent conception.
The source material breaks these down beautifully into distinct mechanistic categories, and we need to master every single one.
Let's guide you through these categories one by one, building a highly structured mental map. The first category is natural or traditional methods. The fundamental governing principle here is incredibly straightforward, preventing the ovum and the sperm from ever meeting,
without the use of any external physical devices or synthetic chemicals.
This category relies entirely on manipulating the timing and the mechanical execution of the reproductive act itself, based on an understanding of the human menstrual and spermatogenic cycles. The first one on the list is periodic abstinence. This is entirely a game of biological timing.
The strategy dictates that couples avoid or abstain from coitus from day 10 to day 17 of the menstrual cycle.
Now, let's connect this back to the endocrinology we studied in Chapter 3. Why that specific 7-day window? Because this is the exact phase when the LH surge, the luteinizing hormone surge, triggers ovulation.
The ovum is released from the Graafian follicle and swept into the fallopian tube.
Biologically, the ovum is only viable and capable of being fertilized for about 24 hours after release. However, human sperm are incredibly resilient. They can survive within the female reproductive tract for up to 3 to 5 days.
Therefore, if ovulation happens on day 14, sperm deposited on day 10 could theoretically still be waiting in the fallopian tube.
And sperm deposited on day 16 might catch a slightly delayed ovum. So this day 10 to 17 window covers all the biological variables. It is the period when the chances of fertilization are at their absolute mathematical highest.
Precisely. By avoiding coitus During that specific physiological window, you are directly mitigating the highest risk period. The second natural method mentioned is coitus interruptus, also commonly known as withdrawal.
The mechanism here is that the male partner actively withdraws his penis from the vagina just prior to ejaculation to avoid depositing semen into the reproductive tract.
While it is one of the oldest recorded methods in human history, it is notoriously unreliable.
From a purely biological standpoint, the pre-ejaculatory fluid secreted by the bulbourethral glands, which is primarily meant for lubrication and neutralizing urethral acidity, can, and very often does, contain viable sperm.
Therefore, insemination can occur even before the primary ejaculation. But it is the third natural method that often catches students off guard and requires a deep understanding of neuroendocrinology.
Yes, lactational amenorrhea. Wait, so it's fascinating when you look at this not just as a medical fact to memorize, but as an elegant evolutionary survival mechanism. So breastfeeding acts as a natural contraceptive.
It absolutely does.
Lactational refers to the intense production of breast milk, and amenorrhea literally means the absence of menstruation. So the act of breastfeeding a newborn actually acts as a systemic, natural contraceptive.
When you think about the energy economics of the human body, it makes perfect sense.
The mother's body is currently expending a massive amount of metabolic energy synthesizing milk to keep a newborn infant alive. The body is essentially calculating its energy reserves and deciding that it absolutely cannot support the immense physiological tax of a concurrent second pregnancy.
It is a brilliant example of systemic hormonal crosstalk. Let's break down the exact neuroendocrine pathway so you truly understand it.
Walk us through it.
Following parturition, the delivery of the baby. If the mother is intensely and exclusively breastfeeding, the physical stimulation of the nipple sends neural signals to the hypothalamus,
Which triggers the anterior pituitary
to constantly secrete high levels of the hormone prolactin, which is essential for milk synthesis. Here's the critical mechanism. These persistently elevated levels of prolactin actively and aggressively suppress the release of gonadotropin-releasing hormone, or GnRH, from the hypothalamus.
And without GnRH, the pituitary cannot release follicle-stimulating hormone and luteinizing hormone.
Without FSH and LH, the ovarian follicles do not develop, there is no estrogen surge, and crucially, there is no ovulation.
Because there is no ovulation, the subsequent menstrual cycle simply does not occur.
So, as long as this intense prolactin-driven lactation continues, the chances of conception are biologically minimized almost to zero.
The body literally halts the reproductive cycle to prioritize the existing infant. It's a flawless piece of biological engineering. But, and this is a critical juncture for anyone taking notes, there is a massive neat trap here regarding the timeline. You cannot assume this suppression lasts indefinitely.
That is correct. Here's what you must remember, and I urge you to underline this in your notes. Lactational amenorrhea is officially recognized as being effective for an absolute maximum period of 6 months following parturition.
6 months.
Examiners will constantly try to trick you with options suggesting it lasts up to a year for the entire duration of breastfeeding or until the infant is weaned. Do not fall for it.
The textbook and medical consensus draw a hard line at 6 months.
Because after that point, the infant usually begins consuming complementary foods, the intensity of suckling decreases, prolactin levels begin to fluctuate, and ovulation can spontaneously return without any warning.
Often before the first postpartum menstrual bleed even occurs. Now, the overarching benefit of all these natural methods is that they carry virtually zero pharmacological side effects, as there are no synthetic hormones or foreign devices introduced into the body.
The significant trade-off, however, is that their overall failure rates are exceptionally high compared to modern clinical methods.
Which naturally brings us to the next phase of human ingenuity. We transition from playing the timing game to erecting a physical blockade. Category 2 barrier methods.
The foundational concept here is simple physical mechanics. The ovum and sperms are prevented from physically encountering one another through the intervention of an impermeable barrier.
Here's where it gets really interesting. We need to highlight a true champion of public health: condoms. They are physical barriers made of very thin, resilient rubber or latex sheaths,
Designed to cover the penis in the male, or the vagina and cervix in the female, immediately prior to coitus.
The physiological result is that the ejaculated semen is entirely trapped within the sheath and physically blocked from ever entering the female reproductive tract.
Condoms stand out in this curriculum, and it's crucial to understand why they are heavily emphasized, not just for family planning, but for global disease control. What is the unique characteristic that separates them from every other method we will discuss today?
Functionally, they are pulling double duty. Condoms are the only contraceptive method highlighted in your syllabus that provides a dual benefit. They are highly effective at preventing conception by trapping the sperm, but crucially, their impermeable latex structure also acts as a physical shield against viral and bacterial pathogens.
They actively protect the user from contracting sexually transmitted infections, STIs.
And specifically the HIV virus leading to AIDS. That dual functionality makes them the absolute cornerstone of public health campaigns worldwide. Furthermore, from a user experience standpoint, both male and female condoms are easily accessible, they are entirely disposable, and they can be self-inserted by the user,
Which guarantees complete privacy.
removing the need for a clinical visit, which heavily drives their widespread social acceptance.
Privacy, accessibility, and disease prevention. Those are the three vectors driving their success. But our mental map of the barrier category isn't complete with just condoms.
Right, there are others.
The curriculum also details diaphragms, cervical caps, and vaults. These are also physical barriers manufactured from rubber or silicone, but their application is different. They are inserted directly deep into the female reproductive tract to tightly cover the external osses of the cervix during coitus,
They act as a physical plug.
locking the primary gateway and preventing the entry of sperms into the cervical canal and the uterus beyond. Now, from an examination perspective, what is the critical distinction a student must remember between these cervical barriers and condoms?
The distinction boils down to the material lifespan and the protocol for use. Unlike condoms, which are strictly single-use, disposable items, diaphragms, cervical caps, and vaults are reusable.
They must be washed and stored properly after each use.
But the real high-yield fact here is how they are deployed clinically. To maximize their contraceptive efficiency and account for any microscopic gaps around the edges of the barrier, they are almost universally paired with chemical agents.
Specifically spermicidal creams, jellies, and foams.
These spermicides contain surfactant chemicals like nonoxynol 9, which actively attack and disrupt the lipid bilayer of the sperm's cell membrane, effectively killing them on contact. So the protocol utilizes a two-front war: a physical rubber barrier blocking the anatomical path, and a localized chemical minefield actively destroying any sperm that managed to get close to the cervix.
An excellent synthesis of the physical and chemical mechanisms. Now, I need everyone to refocus and sharpen their attention because we are entering the ultimate high-yield zone for your board and competitive exams.
Category 3.
Category 3, IUDs or intrauterine devices. If you connect this back to the broader sociological picture of India's population strategy that we opened with, IUDs are arguably the most critical and impactful clinical tool in the nation's arsenal.
They are highly effective, incredibly long-lasting medical devices that must be inserted by qualified doctors or expert nurses directly into the uterus through the vagina.
Once in place, they provide years of uninterrupted contraception, making them one of the most widely accepted and successful methods in India.
Particularly for females who are looking to significantly delay a first pregnancy or dynamically space out the timeline between children. Okay, to master IUDs and guarantee you don't drop any points on these questions, you need to utilize a strict mental map. I like to call it the rule of 3.
3 distinct types.
There are 3 entirely different types of IUDs, and for each one you must memorize the specific textbook examples and, more importantly, the exact biological mechanism of action. Let's break them down systematically. First up, the foundational technology non-medicated IUDs. The classic, highly testable example you need to know here is the lips loop.
Correct. The lips loop is essentially a precisely shaped piece of inert plastic. It does not release any hormones or active chemicals. Its mechanism of action relies entirely on localized physical irritation.
By simply existing as a foreign body occupying space.
Within the delicate environment of the uterine cavity, yes.
Yeah.
It provokes a mild, sterile, chronic inflammatory response in the endometrium. This inflammation triggers a massive influx of immune system cells, specifically macrophages.
These macrophages aggressively increase the rate of phagocytosis of sperms within the uterus.
In simple terms, the immune system treats the sperm as invasive pathogens and literally engulfs and destroys them before they can even reach the fallopian tubes.
So non-medicated equals foreign body inflammation and phagocytosis. Got it. But then we step the technology up a significant notch with the second type, copper-releasing IUDs. Your key textbook examples to memorize here are CUT CU7 and Multilode 375. And the biochemistry here is where it gets really fascinating.
They take that base inflammatory response and add a potent heavy metal toxin to the mix. It is a remarkable application of localized biochemistry.
In addition to the baseline phagocytosis caused by the physical presence of the device, these specific IUDs continuously release microscopic amounts of copper ions into the uterine fluid.
Now, copper ions at this concentration are incredibly highly toxic to human sperm cells. The copper ions permeate the sperm cell membranes and actively interfere with their cellular respiration.
Specifically, the enzymatic processes that generate ATP.
Without ATP, the sperm lose their energy source. This actively and aggressively suppresses sperm motility, meaning the flagella can no longer whip efficiently, and the sperm literally cannot swim.
Furthermore, the copper alters the acrosomal enzymes, severely impairing the fertilizing capacity of the sperms even if they miraculously manage to passively drift near an ovum.
It is a comprehensive biochemical dismantling of the sperm's functional machinery.
So the copper strips them of their energy, paralyzes their motility, and destroys their ability to penetrate the egg. It's a localized chemical warfare zone. That covers the first two. But the third type of IUD is where we see a true systemic biological hack that alters the entire reproductive environment, the hormone-releasing IUDs.
Your specific examples to lock in are Progestisert and LNG20. These are highly sophisticated, precisely engineered delivery systems. Instead of relying on inflammation or heavy metals, they contain a reservoir of synthetic hormones,
Specifically progestogens.
which they release in incredibly small, continuous controlled doses directly into the local environment of the uterine cavity. And here is what you must remember about their mechanism, as it differs entirely from the copper IUDs. They execute two specific, highly effective physiological changes.
That's the first one.
First, they aggressively make the uterus unsuitable for implantation. They act on the endometrial lining, preventing the necessary thickening and vascularization. The endometrium becomes atrophic.
Meaning that even if fertilization somehow occurred in the fallopian tube, the resulting blastocyst would find the uterine wall completely inhospitable and fail to attach.
Second, and perhaps more crucially, they make the cervix intensely hostile to the sperms. The progestogens cause the cervical glands to secrete a thick, highly viscous cross-linked mucus.
This mucus acts as a dense, nearly impenetrable biochemical plug that severely retards and often completely blocks sperm entry from the vagina into the uterus. Unsuitable uterus, hostile cervix. That is the mantra you need to repeat for hormone-releasing IUDs. Progestisert and LNG20 create an environment where sperm cannot enter and an embryo cannot implant. I promise you, understanding that specific mechanistic distinction that copper kills sperm motility while hormones alter the uterine lining and cervical mucus is going to earn you crucial points on test day.
Examiners love to mix those mechanisms up in multiple choice options. Moving along our map, we shift our focus from these localized internal physical devices to systemic, organism-wide chemical hacks. Category 4 covers oral pills, injectables, and implants.
Let's start with the most culturally ubiquitous method, the oral administration of small, precise doses of either progestogens alone or carefully calibrated progestogen-estrogen combinations. These are universally and colloquially referred to simply as the pill.
The logistics and compliance required for taking the pill are very specific and strict. They are formulated to be taken daily for a continuous period of 21 days.
The protocol strongly dictates that a woman should ideally start taking them within the first 5 days of her menstrual cycle. Once the 21 days of active hormone pills are completed, there is a mandatory gap of 7 days.
During this hormone-free week, the sudden withdrawal of the synthetic hormones mimics the natural drop in progesterone, triggering a withdrawal bleed that perfectly coincides with a normal menstruation.
After those 7 days, a new pack is started, and the 28-day cycle is repeated indefinitely. But how do these tiny daily doses of hormones actually prevent pregnancy on a systemic organismal level?
They execute a brilliant manipulation of the body's master control systems, effectively creating a state of pseudo-pregnancy. By orally ingesting these hormones daily, you are keeping the blood serum levels of progestogens and estrogens artificially stable and elevated.
This constant elevation exerts a powerful, continuous negative feedback loop on the hypothalamus and the anterior pituitary gland.
Because the pituitary senses that hormone levels are already high, it drastically inhibits the secretion of follicle-stimulating hormone, FSH, and luteinizing hormone, LH.
If you recall your reproductive physiology, without FSH, no new ovarian follicles mature. Without the mid-cycle LH surge, ovulation is fundamentally inhibited. The ovary simply never releases an ovum.
The factory is essentially put on standby. Furthermore, as secondary defense mechanisms, just like the hormone-releasing IUDs, these oral pills alter the biochemical quality of the cervical mucus to make it thick and viscous.
retarding sperm entry.
And they modify the endometrial lining to inhibit implantation in the extraordinarily rare event an egg is released and fertilized. It's a robust, multi-layered systemic defense.
And when we discuss oral contraceptives in this curriculum, we must pause to highlight a major point of national pride within the Indian scientific and pharmacological community: the development of Saheli.
This isn't just another brand of the standard pill.
It represents a completely different pharmacological approach. Saheli is a highly specialized pill developed entirely by indigenous scientists at the Central Drug Research Institute, the CDRI, located in Lucknow, India.
What makes it so revolutionary is that it is a strictly non-steroidal preparation.
It utilizes a molecule called ormeloxifene, which acts as a selective estrogen receptor modulator, or SERM. Because it isn't pumping the body full of systemic steroids, it only needs to be taken once a week after the initial loading phase.
It boasts a very high contraceptive value while presenting exceptionally few side effects compared to daily steroidal pills.
It is a remarkable achievement in targeted pharmacology that you definitely need to know for the exam.
It certainly is a landmark achievement. Now, the exact same synthetic hormones utilized in standard oral pills progestogens used alone, or in synergistic combination with estrogen, can also be delivered in vastly different formats to eliminate the daily burden of remembering to swallow a tablet.
They can be administered by medical professionals as deep.
Correct. The clinical administration of heavy doses of progestogens or progestogen-estrogen combinations, or alternatively, the rapid insertion of a copper IUD within a strict window of 72 hours of coitus,
have been clinically proven to be highly effective as emergency contraceptives.
The mechanism here is generally a rapid, massive disruption of the ovulatory process, delaying or entirely preventing the release of an egg if it hasn't happened yet.
They are an absolutely vital medical tool for avoiding possible unintended pregnancy resulting from casual, unprotected intercourse, the structural failure of a barrier method like a torn condom, or in the tragic, traumatic instances of sexual assault and rape.
That 72-hour window is the critical parameter for emergency contraception to be effective. The sooner the better.
Okay, we have systematically moved through timing, barriers, localized devices, and systemic chemicals. We have arrived at the fifth and final category of contraceptive strategies in our map. Category 5, surgical methods, which are broadly referred to in the medical literature as sterilization.
These procedures are generally advised and categorized as a terminal method, intended for couples who are absolutely certain they wish to prevent any more pregnancies permanently.
And delving into the anatomy of these procedures brings up a fascinating realization about how the body actually functions post-surgery. When I first studied sterilization, I assumed, like many people do, that it somehow shut down the gonads, that it stopped the body from manufacturing reproductive cells or hormones entirely. But it doesn't do that at all.
No, it certainly does not. The body's biological factories, the testes and the ovaries, keep running perfectly, exactly as they did before the procedure.
Let's look at the specific anatomical interventions.
In the male surgical procedure, which is called a vasectomy, the surgeon makes a small incision on the scrotum, locates the vas deferens, the duct that carries sperm from the epididymis to the urethra, and a small segment of it is surgically removed, and the two cut ends are tightly tied off or cauterized.
In the corresponding female surgical procedure known as a tubectomy, the surgeon accesses the abdomen or the vagina, locates the fallopian tubes, and similarly removes a small segment, tying or clamping the ends to completely sever the connection between the ovary and the uterus.
So if we trace the physiological reality of this, the sheer elegance of the intervention becomes clear. Because the blood supply and nerve connections to the gonads are entirely untouched, the testes are still continuously producing millions of viable sperm every day.
The ovaries are still going through their monthly cycles, maturing and releasing eggs into the abdominal cavity. Furthermore, the endocrine system is completely unaffected. The gonads are still pumping out normal levels of testosterone, estrogen, and progesterone into the bloodstream.
This means the person feels entirely 100% normal. Their libido, their secondary sexual characteristics, their muscle mass, their menstrual cycles, everything remains exactly the same.
But the transport highway has simply been physically severed. The gametes are still diligently produced, but they have absolutely nowhere to go. Because they cannot traverse the blocked ducts, they are eventually broken down and safely, naturally reabsorbed by the body's immune macrophages.
It is a purely structural, highly localized intervention. It permanently blocks gamete transport and thereby mathematically guarantees the prevention of conception. And you must remember a crucial clinical caveat regarding these surgical techniques.
While they are overwhelmingly highly effective, their failure rate approaches absolute zero. Their reversibility is considered to be very, very poor.
While microsurgical recanalization is technically possible in some advanced centers, reconnecting those microscopic delicate tubes years later is incredibly difficult, highly expensive, and very often unsuccessful in restoring actual fertility.
Therefore, it is considered a permanent terminal decision. The selection of this method, or frankly any contraceptive method we've discussed today, should always be undertaken voluntarily and in deep, informed consultation with qualified medical professionals who can explain the long-term implications.
Absolutely. Informed consent is paramount, and as the textbook very astutely points out, we have to remember a fundamental philosophical reality about all of these methods. Contraceptives are not regular biological requirements for the maintenance of an individual's personal reproductive health in the same way that daily vitamins, adequate hydration, or cardiovascular exercise are.
By using contraception, we are actively forcing a medical or chemical intervention against a completely natural, hardwired reproductive event. Therefore, we cannot ignore that there can be real physiological ill effects.
Depending on the method and the individual biology, users might experience nausea, severe abdominal pain, unpredictable breakthrough bleeding, highly irregular menstrual cycles.
Or even, in some statistically rare cases, an increased risk of breast cancer associated with certain hormonal therapies. These risks are real, they must be managed clinically, and they shouldn't be totally ignored or minimized by the medical community.
That's a very mature, necessary clinical perspective to maintain. Okay, let us take a collective breath. We have successfully mapped out the immense architecture of how humanity attempts to pause conception.
We've covered the physics, the chemistry, and the anatomy of contraception. But what happens when those incredibly sophisticated systems fail? Or what happens when a pregnancy occurs, but for severe medical or social reasons, it is simply untenable to continue? That sobering reality transitions us directly into Section 3 MTP and the invisible threat of STIs.
This is a heavy section, both in terms of its medical gravity and its profound societal impact. We begin with the concept of medical termination of pregnancy, or MTP.
By definition, this is the intentional or voluntary termination of a pregnancy before the fetus reaches full term, also commonly referred to in the medical literature as an induced abortion. First, to understand why this is a massive pillar of global reproductive health care, we must grasp the sheer, staggering scale of it.
Statistically, nearly 45 to 50 million MTPs are performed every single year all over the world.
That number is so large it can be hard to visualize, but it accounts for roughly 1/5 of 20% of the total number of conceived pregnancies globally. It is an undeniable massive aspect of human reproductive reality.
Given those sheer numbers, it is obvious that whether to accept, regulate, or strictly legalize MTP is an issue heavily and passionately debated across the globe. These debates are driven by profound emotional, ethical, religious, and complex social issues.
Now, we must apply our strict impartiality rule here clearly. As scientific educators, and basing our discussion purely on the boundaries of your biology textbook and the Indian legal framework, we do not take any philosophical or ethical sides. We are here merely to accurately report the legal statutes and the medical realities provided in your curriculum.
Historically, in India, the government officially legalized MTP in the year 1971. However, they did not do so casually. They enacted it with very strict, specific regulatory conditions.
These conditions were designed specifically to avoid its widespread misuse, particularly aiming to curb the indiscriminate and highly illegal practice of female feticide that we discussed earlier when examining the amniocentesis trap.
Right, the law is designed to thread a very complex needle. So why does the Indian legal framework explicitly allow for MTP? The textbook outlines specific legally sanctioned grounds.
MTP is legally permitted to get rid of unwanted pregnancies that occur due to casual, unprotected intercourse, or, crucially, due to the documented failure of the contraceptive method used during coitus, acknowledging that no method we discussed in Section 2 is 100% perfect.
It is also explicitly permitted in the tragic cases of rape. Furthermore, MTPs are considered legally and medically essential in certain severe cases where the continuation of the pregnancy could be physically harmful or even fatal.
Either to the mother, perhaps due to a severe underlying cardiac or renal condition, or to the fetus, if severe congenital anomalies are detected, or potentially to both.
That legal framework was recently subjected to a critical update, which is highly relevant for your current board exams and Neet preparations. The Medical Termination of Pregnancy Amendment Act of 2017 was enacted by the government with the primary goal of further reducing the rate of illegal, unsafe abortions, and consequently driving down the maternal mortality and morbidity rates that inevitably stem from unqualified quacks performing unsterile, unsafe procedures.
Here is the exact, highly testable legal breakdown you absolutely need to know. If a pregnancy is within the first 12 weeks, which constitutes the first trimester, it may be legally terminated on those aforementioned approved grounds based solely on the medical opinion of one single registered medical practitioner.
That makes sense for early interventions. But what happens as the pregnancy progresses?
The legal requirement shifts significantly. If the pregnancy has progressed past the first trimester, meaning it has lasted more than 12 weeks, but fewer than the absolute upper limit of 24 weeks, the law strictly requires that 2 registered medical practitioners must be consulted.
Both doctors must be of the shared opinion, formed in good medical faith, that the required legal and medical grounds exist to justify the termination. And it is crucial to understand that medically and biologically, there is a very sound physiological reason for this increased legal strictness.
Here's your explicit Neet trap for this section. The textbook states clearly that MTPs are considered relatively safe only during the first trimester, up to 12 weeks of gestation.
Second trimester abortions are exponentially riskier for the mother. Why? We have to look at the embryology and anatomy. By the second trimester, the fetus has grown significantly larger, and more importantly, the placenta has become deeply, intimately integrated into the maternal myometrium, the muscular wall of the uterus.
The vascularization is massive. Attempting to terminate the pregnancy and separate those tissues at that advanced stage poses severe, life-threatening risks of massive hemorrhage, uterine perforation, and complex post-operative complications for the mother.
That biological reality perfectly explains the legal distinction. The law reflects the anatomy. First trimester requires 1 doctor because it's safer. The 12 to 24-week window requires 2 doctors because the physiological risks to the mother skyrocket.
Okay, shifting gears from MTP, we need to address the second major component of this section, a massive, silent threat to a reproductively healthy society. We are talking about sexually transmitted infections, or STIs. In older literature or varying questions, you might also see them referred to as venereal diseases or reproductive tract infections. They all refer to the same category of pathogens.
The curriculum lists several specific pathogens and the diseases they cause that you need to be familiar with. The list includes bacterial infections like gonorrhea and syphilis, viral infections like genital herpes and hepatitis B, parasitic infections like trichomoniasis, and other bacterial issues like chlamydiasis.
It also includes genital warts caused by HPV and, of course, HIV leading to AIDS.
Now, let's identify the absolute highest yield, most frequently tested fact in this entire segment. I call it the Incurable Three. Here is what you must remember, and examiners love testing this distinction. Out of that entire list, hepatitis B, genital herpes, and HIV are not completely curable.
Let me repeat that clearly for the listener taking diligent notes, because it is that important. Hepatitis B, genital herpes, and HIV are not completely curable. Every single other infection on that syllabus list, gonorrhea, syphilis, chlamydia, trichomoniasis, is completely curable, provided it is detected early and treated properly with a full course of specific antibiotics or antiparasitics.
But those 3 specific viral infections integrate into the body in a way that makes them lifelong conditions. They can be managed with antiviral therapies, but the pathogen cannot be entirely eradicated from the host.
Exactly. The virology dictates the incurability, and another crucial concept to remember here is the overlap in transmission routes. By definition, most of these diseases are spread primarily or almost exclusively through intimate sexual contact with an infected partner.
However, hepatitis B and HIV are unique, highly dangerous outliers. They are blood-borne pathogens, which means they can also be efficiently transmitted by sharing injection needles among intravenous drug users by utilizing unsterilized surgical or dental instruments through the direct transfusion of infected blood products.
Or vertically, meaning transmitted directly from an infected mother to her developing fetus across the placenta or during childbirth. The transmission overlaps are a public health nightmare. But here is the truly mind-blowing realization regarding STIs, and it's terrifying because it highlights their evolutionary biological stealth.
Why are these specific infections so wildly successful at spreading through human populations?
It is precisely because their early symptoms, if there are any symptoms at all, are incredibly minor and easily dismissed. We are talking about extremely subtle signs: slight localized itching, a minor increase in fluid discharge, a vague sense of minor pain, or slight swelling in the genital region.
And crucially, the textbook explicitly points out that infected females are very often entirely asymptomatic in the early stages. The anatomy of the female reproductive tract can hide these minor inflammations deep inside.
So a person can be highly infectious, carrying a dangerous pathogen, and have absolutely zero physical idea that anything is wrong.
This profound biological silence is the true danger. When you combine this asymptomatic silent reality with the intense, highly prevalent social stigma attached to sexually transmitted diseases in almost all societies, you create a perfect storm for an epidemic. What happens? Even if someone notices a minor symptom, they are often deterred from going to a doctor for timely detection and proper simple treatment.
They actively ignore the minor symptoms, hoping they will disappear, driven entirely by fear, embarrassment, or social shame. And the physiological consequences of that delay, driven by stigma, are absolutely devastating. Because the infection doesn't just magically go away, it uses that time to move deeper and higher into the reproductive tract.
This silent progression leads directly to severe late-stage complications, the most prominent being pelvic inflammatory diseases, or PID.
PID is a severe, widespread infection of the female reproductive organs, moving from the cervix up into the uterus and the fallopian tubes. It can cause horrific damage. It leads to spontaneous abortions, tragic stillbirths, and incredibly dangerous ectopic pregnancies.
Let's quickly define why ectopic pregnancies occur in this context, as it's a great physiological connection. The chronic inflammation from PID severely damages and scars the delicate ciliated epithelium lining the inside of the fallopian tubes.
Without those beating cilia to sweep the fertilized zygote down into the uterus, the embryo gets stuck and implants right there in the narrow fallopian tube.
As it grows, it eventually ruptures the tube, causing massive, life-threatening internal bleeding.
That is a terrifying anatomical cascade. And ultimately, if left untreated, the severe scarring from STIs and PID can lead to permanent, irreversible infertility. In some cases, like with specific strains of HPV causing genital warts, it can directly trigger cancer of the reproductive tract, particularly cervical cancer. So a minor, ignored infection turns into a lethal oncology case.
This is exactly why STIs are considered a paramount threat to a healthy society. And I want to pause and speak directly to the listener here, stepping out of the textbook for a moment. The demographic statistics are stark and clear. Incidences of these infections are reported to be overwhelmingly high among persons in the age group of 15 to 24 years.
Given that you are studying for medical entrance exams, that is very likely the exact demographic age group you belong to right now.
But the text is also clear on the solution. There is no reason to panic, because prevention is entirely practically possible. The rules of public health are simple, effective, and protective. Strictly avoid sex with unknown partners or multiple partners. Always try to use condoms, which provide that essential physical latex barrier against pathogens. And crucially, in case of any doubt or symptom whatsoever, immediately seek out a qualified, non-judgmental medical doctor for early detection and rapid treatment. Do not ever let temporary social stigma cost you your long-term permanent health.
That is such a vital message, both for scoring well on the exam and for navigating life itself. And the mention of STIs ultimately leading to irreversible infertility brings us perfectly and logically to our final major topic of the day. Section 4, Engineering Life. We are going to deeply explore infertility and the marvels of assisted reproductive technologies, or ART-T. Let's start with establishing a firm clinical baseline. If an examiner asks for the exact technical definition of infertility, what are the strict parameters a student must provide?
The exam-ready clinical definition is highly precise. Infertility is defined as the inability to successfully conceive or produce children in spite of 2 years of continuous, unprotected sexual cohabitation. The timeline there is the critical metric. It is not 6 months of trying, it is not 1 year. Clinically, the threshold is 2 full years.
And the underlying reasons for this inability can be incredibly varied and complex. It could be gross physical abnormalities, severe congenital diseases, the disruptive side effects of certain necessary pharmaceutical drugs, complex immunological reactions where the female's body literally attacks and rejects the male's sperm as foreign invaders, or even deep-seated complex psychological factors impacting sexual function. But beyond the biology, there is a profound, recurring social observation explicitly made in the textbook that I think is incredibly important to highlight.
The text notes that in India, there is a pervasive, deeply unfair social tendency where the female partner is almost automatically and exclusively blamed for a couple being childless. But the textbook clearly and forcefully states that, medically speaking, more often than not, the physiological problem actually lies within the male partner.
Yes, that is a perfect example of why rigorous scientific education is necessary to dispel harmful, unscientific social myths. Male factor infertility is massive. It could be oligospermia, a drastically low sperm count, or asthenozoospermia, which is poor sperm motility where the sperm simply cannot swim straight or fast enough, or structural inability to inseminate. These are massive, common physiological factors that are far too often socially ignored or deflected.
Now, specialized healthcare units, such as dedicated infertility clinics, possess the diagnostic tools to often identify and treat these underlying disorders, perhaps with hormone therapy or minor corrective surgeries. But when those initial corrective treatments fail or aren't physically possible, modern medicine steps in with an array of staggering, high-tech interventions known collectively as assisted reproductive technologies, or RT. This is truly the frontier where precision human engineering meets human biology.
Okay, let's build a comprehensive learner's guide to ART. When you first look at this section, it can look like a confusing alphabet soup of acronyms. IVF, ZFT, IUT, GIFT, ICSI can be overwhelming. But we can make it crystal clear by organizing them based on a very simple, logical flow. Ask yourself one question: where does the actual microscopic moment of fertilization happen? Let's start with the scenarios where fertilization happens entirely outside the human body. That brings us to the pioneer of the field, in vitro fertilization, commonly known as IVF.
The Latin term in vitro literally translates to in glass, referring to laboratory glassware. This is the scientific process behind the famous colloquial term test tube baby program. The procedure is complex. Multiple mature ova from the wife or a rigorously screened donor female and a concentrated sample of sperms from the husband or a donor male are collected. They are then brought together in highly simulated, pristine laboratory conditions.
The lab must perfectly mimic the internal environment of the fallopian tubes, matching the exact pH, temperature, and osmolarity. Under these perfect conditions, the sperm are induced to penetrate the ovum and form a zygote right there in the petri dish.
But obviously, a human embryo cannot grow to term in a plastic petri dish. It has to go back into the maternal environment. This crucial secondary step is called Embryo Transfer, ET. And listen carefully, because here is your massive, undeniable, neat trap alert for the entire ART section. The examiners will relentlessly test you on exactly where the embryologist chooses to transfer that embryo, and that decision is based entirely on the embryo's cellular stage of development. You have to literally count the blastomeres.
The individual cells of the rapidly dividing early embryo?
Here's what you must remember, and you must lock this logic into your brain. The magic deciding number is 8. If the lab has cultured a zygote, or an early embryo, that is divided up to and including exactly 8 blastomeres, it is surgically transferred high up into the fallopian tube of the female. This specific procedure is called ZIFT, zygo-intrafallopian transfer. Now, why the fallopian tube?
Because we rely on chronological and biological synchrony. In a completely natural, unassisted pregnancy, an embryo at the tiny 8-cell stage is exactly physically located in the fallopian tube, slowly tumbling its way down. It requires the specific biochemical secretions of the tubal epithelium to survive and continue its cleavage before it is ready to handle the uterine environment.
Exactly. It's about putting the right stage of embryo into the right anatomical environment. But if the embryologist decides to let the embryo culture in the lab for a few more days, and it develops into a more advanced stage with more than 8 blastomeres moving towards the morula or blastocyst stage, it has fundamentally progressed past the tubal stage of its life. If you put it in the tube now, it might cause an ectopic pregnancy. It is now biochemically ready for the thick lining of the uterine environment.
So embryos with more than 8 blastomeres are transferred directly deep into the uterus to complete their development and attempt implantation. This procedure is called IUT, intrauterine transfer. The rule is absolute. Up to 8 cells equals ZIFT into the tube. More than 8 cells equals IUT into the uterus. Lock that distinction into your memory. It is guaranteed points.
Now let's shift our criteria. What if the actual moment of fertilization happens inside a female, naturally, but the resulting embryo is then carefully recovered and transferred out to assist another female? This is classified as in vivo fertilization. In vivo meaning within the living body. Embryos successfully formed by the natural fusion of gametes within a fertile female can be safely flushed out and transferred to assist a woman who perhaps cannot conceive an embryo herself, but can successfully carry a pregnancy to term.
Okay, so that covers transferring the embryo itself. But what if we move back a step? What if a woman's reproductive problem is strictly ovarian? She cannot produce a viable ovum due to premature ovarian failure or severe structural issues, but her fallopian tubes are clear and her uterus is perfectly healthy and entirely capable of carrying a baby. This specific scenario is where GIFT comes into play, gamete intrafallopian transfer.
Correct. The name tells you exactly what is happening. In GIFT, you're not transferring an embryo. You are transferring unfertilized gametes. Specifically, a surgeon collects a healthy unfertilized ovum from a generous donor. This ovum is then transferred directly into the functional fallopian tube of the recipient female, who cannot produce her own eggs. The key physiological requirement here is that this recipient female must be able to provide a suitable, healthy internal environment for natural fertilization to occur within her own tubes and for the subsequent embryonic development to proceed normally in her uterus.
The analogy I always love using for GIFT is agriculture because it makes the rules so clear. Imagine if you are a farmer who owns an incredibly rich, highly fertile plot of soil. The climate is perfect, the irrigation is flawless, but you simply don't have any seeds to plant. The soil is useless without the seed. In this scenario, GIFT is the specialized doctor stepping in, providing the high-quality seed, which is the donor ovum, and carefully placing it right into your perfect soil, the healthy fallopian tube. Once the seed is placed, natural fertilization with the partner's sperm, which happens through normal coitus or artificial insemination, can then take place entirely inside the body, naturally.
A very apt and visually helpful analogy. Now, if we connect this all back to the broader picture of pushing the absolute limits of high-precision biological engineering, we must examine ICSI intracytoplasmic sperm injection. In standard, traditional IVF, the embryologist simply places the extracted egg and thousands of prepared sperms together in a dish and relies on nature to take its course. They hope that at least one sperm will have the motility to swim to the egg and the acrosomal enzymes to digest the zona pellucida and penetrate it on its own. But what if the male partner's sperm are completely immotile or morphologically severely abnormal, preventing them from doing this? In ICSI, the embryologist uses incredibly fine microscopic glass pipettes under a high-powered microscope to physically capture one single visually viable sperm. They then puncture the egg's protective layers and directly inject that single sperm straight into the cytoplasm of the ovum. It is the ultimate brute force circumvention of any and all natural sperm motility or penetration issues. It is stunning, microscopic precision. They are literally forcing fertilization to happen cell by cell. And speaking of male factor infertility, there are less invasive, simpler procedures available if the male partner simply has a very low sperm count or is structurally unable to inseminate the female naturally. This approach is artificial insemination, or AI. In this procedure, a semen sample is collected either from the husband or a rigorously screened healthy donor. The sample is often washed and concentrated in the lab to isolate the healthiest, fastest sperm, and then it is artificially introduced deep into the vagina.
If the doctor uses a fine catheter to introduce that concentrated semen directly deeper into the uterine cavity, completely bypassing the potentially hostile environment of the cervical mucus we talked about earlier, it is specifically called IUI, intrauterine insemination. This drastically shortens the distance the sperm have to swim and ensures a high concentration reaches the fallopian tubes.
It truly is a remarkable, almost science fiction-like suite of medical technologies. However, the textbook brings us firmly and necessarily back to stark reality at the end of this chapter. These RT techniques are not magic bullets, and they are not universally accessible. They require extremely high precision handling by highly specialized, extensively trained medical professionals. They require immensely expensive, delicate instrumentation, sophisticated incubators, micromanipulators, cryogenic freezing equipment. Because of this, they're currently only available in a few highly specialized urban centers across the country, and the sheer financial cost makes them affordable only to a very limited, privileged demographic of people. Furthermore, the emotional toll of repeated failed cycles, along with complex religious, ethical, and social factors, act as very strong deterrents for many couples considering these intensive methods.
And that stark reality leads directly to a really beautiful, profoundly human concluding thought from the source material. Since the ultimate driving aim of all these complex procedures, all this massive medical expense, and all this agonizing emotional stress is simply the deep human desire to have children to love, to care for, and to raise. We must acknowledge that we already have so many orphaned and destitute children in India. Children who are already here who probably would not survive to a healthy maturity unless they are taken in and lovingly cared for by a stable family. The textbook explicitly and purposefully highlights that our national laws permit and encourage legal adoption, and it remains one of the absolute best, most practical, and most deeply compassionate methods for couples looking to experience the joy of parenthood. You don't always need a micromanipulator to build a family. Sometimes you just need an open heart and an open home.
It is an incredibly profound and necessary note to end the clinical chapter on. It acts as a perfect full circle, reminding us that reproductive health, family building, and human biology ultimately tie directly back to the social, emotional, and behavioral well-being of the entire community, aligning flawlessly with the four-pillared WHO definition we started with an hour ago.
All right, my friend, we have covered an absolutely immense amount of scientific and historical ground today. We have traversed post-independent sociology, demographic mathematics, reproductive anatomy, systemic pharmacology, and high-tech micromanipulation embryology. It is time to lock this vast web of knowledge in for good. Let's move to our outro. We need to deploy those memory anchors to make sure these highly testable concepts survive the stress of exam day.
Let's solidify this foundation. I want you to focus intently right now. Here's what you must remember. These are the 5 critical, non-negotiable facts you absolutely cannot afford to forget or confuse from Chapter 4. Number 1, the amniocentesis trap. Amniocentesis is a fetal cell test utilizing karyotyping that is legally banned in India specifically for sex determination in order to combat the social evil of female feticide. It is not banned for identifying genetic chromosomal disorders.
Number 2, the timeline of nature. Lactational amenorrhea acts as a powerful natural contraceptive by suppressing GnRH and ovulation through high prolactin levels, but it is clinically reliable for an absolute maximum of only 6 months following parturition. Do not guess any higher.
Number 3, the IUD mechanisms. Hormone-releasing IUDs, specifically progesterone and LNG20, work systemically by making the endometrium unsuitable for implantation and making the cervical mucus intensely hostile and thick against sperms, whereas copper IUDs suppressed sperm motility.
Number 4, the virology of STIs. Remember the Incurable Three. Hepatitis B, genital herpes, and HIV are viral infections that are not completely curable, whereas bacterial infections like gonorrhea and syphilis are entirely curable if detected early.
Number 5, the embryology math of RT. When looking at IVF transfers, count the blastomeres. Less than or equal to exactly 8 blastomeres dictates ZIFT transfer into the fallopian tube. Anything more than 8 blastomeres dictates IUT transfer directly into the uterus.
Boom. Those 5 anchors are pure, high-yield gold. Do not walk into the exam hall without them memorized. Now let's bring this massive topic completely together. Give me your focus for the 60-second sticky summary. If you somehow forget everything else from this hour, take this core narrative with you. Reproductive health isn't just about anatomical plumbing. It is the total physical, emotional, behavioral, and social well-being of a population. Because our medical successes drastically dropped mortality rates, the global population exploded mathematically, forcing humanity to try and artificially control our biological drive. We counter this drive with natural timing based on the menstrual cycle, physical barrier methods like condoms, which pull double duty by blocking both sperm and deadly STIs, chemical hormones and pills and IUDs that trick the pituitary into not ovulating or altering the uterine environment, and permanent surgical blocks that sever the transport highways without altering hormones. When contraception fails, MTP serves as a highly regulated medical safety valve, strictly safer only up to 12 weeks of pregnancy. Meanwhile, STIs lurk as a silent, heavily stigmatized, often asymptomatic threat that, if ignored, cause severe, permanent long-term damage like pelvic inflammatory disease, ectopic pregnancies, and infertility. And finally, when human nature fails to conceive after 2 full years of trying, RT steps in with incredible microscopic precision like ZIFT, IUT, and ICSI to engineer fertilization, although the profoundly compassionate act of legal adoption always remains a beautiful, highly practical alternative for building a loving family.
A flawless, rapid-fire synthesis of the entire curriculum. You now possess the complete blueprint.
So what does this all mean for us moving forward? We've looked at the hard facts, the underlying biochemical mechanisms, and the strict legal frameworks governing reproduction today. But as we wrap up this deep dive, I want to leave you with something to really mull over, a thought that pushes past the boundaries of your current textbook.
I think the most provocative, almost unsettling idea hidden just beneath the surface of this text is the relentless paradox of medical progress. We spent significant time discussing how our initial medical successes in saving lives, plugging the drain of mortality, actually led to the massive population explosion. Our sheer success inadvertently created a massive existential sociological crisis regarding planetary resources. But look at where we ended up in the final section. with assisted reproductive technologies. Look at the trajectory. We are already actively manipulating gametes outside the body in simulated petri dish environments. We are routinely bypassing the natural environment of the fallopian tubes entirely. We're using microscopic needles to inject a single chosen sperm directly into an egg, completely circumventing natural selection at the microscopic level. As we continue to aggressively push these boundaries of art, and as biological science steadily moves toward theoretical concepts like ectogenesis, the development of entirely artificial wombs, capable of gestating a fetus outside the human body, we have to ask ourselves a profound question. Will we eventually, inevitably, reach a point in the future where human reproduction is entirely, completely decoupled from human biology? If babies are conceived in labs and gestated in artificial environments, will the physical pillar of reproductive health that the WHO defined become entirely obsolete? Will reproduction become an act of pure engineering rather than biology? It was a staggering ethical, sociological, and scientific question that redefines what it means to be human. It is something deep and slightly unnerving for you to ponder as you close your books today.
What a monumental, perspective-shifting thought to end on. It truly makes you realize that the biology you are studying today is just the stepping stone for the science fiction of tomorrow. Thank you so much for joining us at the table today, for engaging with the material, and for letting us guide you through this complex landscape. Keep that sharp curiosity alive, review those 5 memory anchors until they are second nature, and we will absolutely see you next time as we continue to dive deep into the endlessly fascinating machinery of life.